Hi, the title of today's lecture is Natural Selection. What I'm going to do now is kind of switch gears a little and leave the cellular and molecular basis of evolution behind and return to the theory of natural selection. Return to the theory of evolution, that is. Previously we spoke about some of the major players in the field of natural history and evolution, and we spoke about these individuals from the 16th through the 19th century. We learned about the contributions of Mendel, Darwin, Malthus, Newton, just to name a few. What I'd like to do now is I want to introduce what the major developments were during the 20th century, the major developments to the theory of evolution and natural selection. By the start of the 20th century, the essential foundations for evolutionary theory had already been developed. Darwin had already articulated the key theory of natural selection 40 years earlier, and there was the rediscovery of Mendelian genetics in the 1900s. And this contributed another very important component to our understanding of natural selection and evolution, because it provided a mechanism for inheritance and maintaining variation. We might expect that a synthesis of these two basic contributions would have been joined together relatively quickly into a consistent theory of evolution, but that was not the case. A synthesis of these two ideas did not occur until the 1930s. Okay? Sorry. During the 1930s, biologists who were studying mathematical models of evolutionary change came to realize that small new changes in genetic material that are transmitted from parent to offspring by strict Mendelian principles represent the fuel for natural selection. Let me say that again. Small changes in the genetic material represent the fuel for natural selection. Those new changes that are in the genetic material are transmitted from parent to offspring by Mendelian principles. This idea brought together the two major fields in evolutionary thinking, and together they comprised what is called the modern synthesis. So Darwinian and Mendelian evolution. The basic idea behind the modern synthesis is that evolution occurs in two stages. The first stage is the production and redistribution of variation. That is, inherited differences between individuals is what that is how variation is produced and redistributed. The second phase of the modern synthesis is that natural selection then has the opportunity to act on those inherited differences. It either acts on the inherited differences or it acts specifically on the variation among individuals that differentially affects their ability to reproduce. I want to make it clear that natural selection is the evolutionary force that causes changes in allele frequencies in populations due to differential net reproductive success of individuals. That is, natural selection is what causes changes in the allele frequencies in populations, and it causes it because of differences in the reproductive abilities of individuals due to those changes, or differences rather, in those allele frequencies. So before, when I first introduced evolution, we defined evolution as change over time. Following the modern synthesis, evolution is no longer defined as change over time, but evolution is now defined as a change in the allele frequency from one generation to the next. These allele frequencies are effectively numerical indicators of the genetic makeup of a population. Recall that a population is an interbreeding group of individuals. I also want to remind you that populations evolve, not individuals. So individuals don't evolve, populations do. And I want to also remind you that alleles are one of several forms of the same gene. So let's look at an example of changing allele frequencies. Effectively, that means evolution. We're looking at an example of evolution. And we're going to look at the ABO blood group. Okay? Let's assume that we know all the blood types of all the individuals in a classroom. and that this classroom represents a population of interbreeding individuals. Just to remind you, to be a population of interbreeding individuals, the individuals in this classroom must be more likely to choose a mate from within this group than from outside it. Let's say that Texas A&M Aggies are more likely to choose a mate from who's also a Texas A&M graduate or alum than they are to choose one from that other Texas university. Let's say that the proportion of each of the alleles in the classroom are the following. Let's say there are 30 individuals in the classroom, 15, 12, 3 equals 30. Yes. Okay. Just checking my math. And we have 3 types of blood in this classroom. We have A blood, we have B blood, and we have O blood. We have 15 individuals in the class that have A blood. We have 12 individuals who have B blood, and we have 3 individuals that have O blood. What we now need to calculate the proportion of alleles in the classroom. The proportion of A alleles is 15 divided by 30, the total number of alleles in the class, which equals 0.50. B equals 12 divided by 30, which is 0.40, and the proportion of O alleles is 3 divided by 30, which is 0.10. So notice that the proportion of all these alleles equals 1, 0.5, 0.4, 0.1 equals 1.0. Let's say, God forbid, that I am teaching this class in 25 years, and that the same students are in this classroom 25 years from now, so none of us have moved on. The proportion of the alleles 20 and 25 years might have changed. The proportion of the alleles might be, we still have 30 individuals in the class, but the proportion of alleles is 9 individuals with A blood, 9 divided by 30 equals 0.3, 12 individuals with B blood, so that hasn't changed, and 9 individuals with O blood, 9 divided by 30 is 0.30. So the proportion of alleles over time has changed. During this 25-year period, we see that the relative proportions of A, B, and O blood have changed. A has decreased, O has increased, and B has remained the same. Over the short term, these changes in allele frequencies are relatively small, but if they are continued and elaborated over long periods of time, they can and do produce spectacular types of evolution. I want to make it very clear that we say that a population is evolving if frequencies of its alleles are changing. So in the previous example, we say that the population is evolving because the allele frequencies have changed. If the population is not evolving, the allele frequencies are going to be constant, and the population is in what we call a genetic equilibrium. There are 2 main types of evolution that I want to talk about. The first is called microevolution, the second is macroevolution. Microevolution constitutes short-term evolutionary changes. Macroevolution are long-term changes throughout fossil history. So when we're looking at changes in allele frequencies, we're talking about microevolution. When we're talking about the evolution of dogs from wolves, we're talking about macroevolution. So there are 5 major causes of changes in allele frequencies. The 5 causes of changes in allele frequencies are the 5 evolutionary forces. These evolutionary forces are natural selection, mutation, migration, genetic drift, and non-random mating. These are the 5 evolutionary forces that cause changes in allele frequencies, that cause evolution. So all 5 of these forces cause evolution to occur. Natural selection, mutation, migration, genetic drift, and non-random mating. I'll start out with a discussion of natural selection. Now, we know all about natural selection from Darwin. Natural selection effectively filters genetic variation. Individuals that have certain biological characteristics that allow them to survive and reproduce are more likely to pass on their alleles for those characteristics that allow them to survive and reproduce to the next generation. Natural selection, very importantly, does not create novel genetic variation. Natural selection does not create genetic variation. It only changes the relative frequencies of the different alleles. Okay, so it's a very important point. What we're going to see is that mutation is the only evolutionary force that creates variation. Natural selection acts on that variation. It modifies the frequency of that variation. The analysis of natural selection focuses on what we call fitness. Fitness is the probability of survival and reproduction of an organism. It's the proportion of individuals with a genotype that survive and reproduce. There are two basic components of fitness. The first component of fitness revolves around differential survival or mortality. In certain individuals have greater survival and mortality. Secondly, certain individuals have greater fertility. That is, they produce more offspring or more offspring that survive to reproduce themselves. So let's imagine a locus with the genotypes with two alleles, okay? Dominant allele and recessive allele, and it has three genotypes: homozygous dominant, heterozygous, and homozygous recessive. All the individuals that are heterozygous and homozygous dominant individuals survive and reproduce, and only half the individuals that are homozygous recessive survive. This means that the homozygous recessive genotype has half the fitness of the homozygous dominant and the heterozygous genotype. Fitness refers to the proportion of individuals with a genotype who survive and reproduce. The fitness is always measured relative to the other genotypes. It's never something unto itself. Okay? Let's say we have an example, and we have individuals with the homozygous dominant genotype have 4 kids, individuals that are heterozygous have 2 kids, and individuals that are homozygous recessive have 1 child. Each genotype has a fitness relative to the other genotypes. The genotype with the highest fitness equals 1. So the homozygous dominant genotype, where they have 4 kids, which is the maximum number, has a fitness of 1. The heterozygous genotype has half the number of kids of the maximum number of kids that are produced in this population, and so they have a fitness of 0.5. The homozygous recessive genotype has 1 child out of the maximum of 4 children, so it has a fitness of 0.25. So the fitness is always relative to the other genotypes. Fitness is usually abbreviated mathematically as W. The genotype is written as W1, W2, or W3 based on the number of different types of genotypes in the population. So in this example, the homozygous dominant genotype will have a fitness of W1. The heterozygous will have a fitness of W2. The homozygous recessive genotype will have a fitness of W3. Genotypes also have what we call a selection coefficient. The selection coefficient is generally written as S, and the selection coefficient is always the inverse of fitness. Okay, the selection coefficient is the inverse of fitness. So each genotype the selection coefficient is written as S1, S2, or S3. The homozygous dominant is written as S1, the heterozygote as S2, the homozygous recessive as S3. Fitness plus the selection coefficient equals 1. W plus S equals 1. So fitness plus the selection coefficient equals 1. We say that the selection coefficient S1 for the homozygous dominant genotype equals 0 because the fitness of population 1 is 1. The selection coefficient for the heterozygous genotype is 0.5 because fitness for population 2 is 0.5. We say the selection coefficient for the homozygous recessive genotype is 0.75 because fitness for population 3 is 0.25. keep in mind that there's selection for a genotype or an allele when it has a higher fitness. There is a selection against a genotype or allele when it has a lower fitness. For a single trait, we generally observe 4 modes of selection: against the recessives, against dominant alleles, against homozygous genotypes, and against heterozygous. And we're going to talk about these 4 modes of selection. So let's first start with looking at the mode of selection called selection against the recessive alleles. Let's consider what happens when one allele is dominant and one allele is recessive. Let A be the dominant allele and recessive A be, or lowercase a, be the recessive allele. Based on what we have learned so far, the genotypes homozygous dominant and heterozygous will both give rise to the same phenotype because dominant A is dominant. Because homozygous dominant and heterozygous specify the same phenotype, they are going to have the same fitness. So these individuals here are going to have the same fitness as this individual here. In this example, let's say that the fitness of the homozygous dominant and the heterozygous is 100%. That is, all the individuals that are purple have 100% fitness, and the fitness of the homozygous recessive individual, this individual down here, is 0. And what this means is that no individuals that are homozygous recessive survive to reproduce, and that the homozygous recessive condition is fatal. Okay, so this is how I like to write it out so that it flows better, I think, in your notes and other places when you write it out in this manner. This is the original population. We have the genotype, the fitness. N is the total population size. A is the frequency of the dominant allele, and recessive A here is the frequency of the recessive allele. We start out with just the genotypes, and we know that the fitness of the homozygous dominant and the heterozygous is 1. And we know that the fitness of the homozygous recessive is 0. And we have 50 individuals that are homozygous dominant, 100 individuals that are heterozygous, and 50 individuals that are homozygous recessive. Now we have a total of 200 individuals in the population. The first thing we need to do in order to determine how selection is operating against this recessive allele, or how it affects the allele frequencies when selection is operating against a recessive allele, and the homozygous recessive genotype has a fitness of 0, but the heterozygous is still maintained in the population, we have to first calculate the allele frequencies, the frequency of the dominant and the recessive allele in these populations. So we start out, individuals with the homozygous dominant genotype there are 50 of them, and they each have two dominant alleles, so 50 times 2 is 100. They have no recessive alleles. The heterozygotes, there are 100 individuals. They have 100 dominant alleles and 100 recessive alleles. The homozygous recessives, there are 50 individuals. They have no dominant alleles, and since they have two recessive alleles, 2 times 50 equals 100. So the frequency of the dominant allele is 100 plus 100 plus 0 is 200. 200 divided by the total number of alleles in the population, which is 400. So 200 divided by 400 equals P equals 0.5. Q is for the recessive allele. 0 plus 100 is 200. 200 divided by the total number of alleles in the population, 200 plus 200 is 400. So 200 divided by 400 equals Q equals 0.5 also. So here we did it. A equals the allele frequencies. There were 200 dominant A alleles, 400 total alleles. P equals 0.5. A is the allele frequencies. There are 200 recessive alleles out of 400 total alleles in the population, so Q equals 0.5. So let's look at what happens after selection after one generation of selection against the recessives. We said that the homozygous recessives have a fitness of 0. So in the next generation, there are going to be no individuals. So instead of having 200 individuals in the population, we now have 150. So we still calculate our alleles. The frequency of the dominant alleles are 100, because 50 times 2. The frequency of the heterozygotes is 100 and 100, so we have 300 total alleles in the population. The frequency of the dominant allele is now 200 divided by 300. The frequency of the recessive allele is now 100 divided by 300. So what does this amount to? Then now we have the frequency of the dominant allele is 200 dominant A alleles divided by 300 total alleles. The frequency of P is now 0.667. The frequency of the recessive allele, we have 100 recessive alleles from the heterozygotes, even though the homozygous recessives were all eliminated. And we have 300 total alleles, so the frequency of Q is 0.333. So after selection against the homozygous recessive genotype, there are only 150 individuals, and the allele frequencies have changed. This demonstrates the effect of selection against a homozygous recessive genotype. The frequency of the recessive A allele has dropped from 0.5 to 0.333. The recessive A allele is harmful in the homozygous form, and as such, the fitness for in the homozygous form in that it was 0 and it was eliminated. However, the recessive A allele is not going to be eliminated completely from the population, and this is because the heterozygote condition is not eliminated through selection, because that has a fitness of 1 because of the dominant allele. So the A allele, the recessive A allele, can continue to be maintained in the population, and it is not going to be eliminated from a single generation. Okay? The frequency of the recessive A allele does not decrease at the same rate over time. In fact, it slows down. The amount of reduction in the recessive A allele slows down over time, and as the frequency of the recessive A approaches 0, an increasingly lower percentage of the allele frequency will be homozygous recessive. That is, fewer individuals are going to be eliminated every generation. So, however, that said, the reduction in the recessive A is going to be offset by new mutations from dominant to recessive A. The mutations are slow, so the frequency of the recessive A will only be slightly greater than 0. And ultimately, a balance will be reached as the reduction in the recessive A due to selection is offset by new mutations from dominant to recessive A. So the effects of natural selection, I want to make it very clear, are dependent on both the initial allele frequencies, whether an allele is dominant or not, and the exact values of fitness for each genotype, not just for one genotype. Okay, let's look at another example against homozygous recessive genotypes. So we have our 3 genotypes: homozygous dominant, heterozygous, and homozygous recessive. We have 3 fitnesses. Homozygous dominant and heterozygous still have a fitness of 1, but in this case, instead of having the homozygous recessives having a fitness of 0, well, they have a fitness of 0.75, just slightly less than the other genotypes. The selections are thus: if you have a fitness of 1, selection coefficient 0. Fitness of 1, selection coefficient of 0. Fitness of 0.75, your selection coefficient is 0.25. These are your genotype frequencies, okay, for the population: 0.36, 0.48, and 0.16. The first thing we need to do is calculate the allele frequencies for the dominant and the recessive alleles. The frequency of the dominant allele equals 0.36. This is the frequency of the homozygous dominant genotype, plus this is the frequency of the heterozygote, and we divide it by 2 because they have both alleles, and we get 0.6. The frequency of the recessive allele is 0.16. That's the frequency of the homozygous dominant genotype, plus half the frequency of the heterozygote gives you 0.4. The question we now ask is, what is the loss in genotype and allele frequencies in this population due to selection? Selection against the homozygous recessive genotype. Okay? To calculate the loss due to natural selection, what we do is we multiply the selection coefficient S by the genotype frequencies. So here we have the genotypes, we have our S, our selection coefficients based on calculated from our fitness, and we have our genotype frequencies. The first thing we do is we multiply S by the genotype frequencies. 0 times 0.36 equals 0. 0 times 0.48 is 0. So you'll see there's no loss in the homozygous dominants, there's no loss in the heterozygotes, because we're looking at selection against homozygous recessive. Now we have 0.25 times 0.16, and it gives us a loss of 0.04. Okay, so this is the loss in the homozygous recessive genotypes. So what happens after one generation of selection against the recessive homozygous genotype? We have 3 genotypes. We have the homozygous dominant, the heterozygote, and the homozygous recessive. The original frequencies of the homozygous dominant was 0.36, the original frequency of the heterozygote was 0.48, and the original frequency of the homozygous recessive was 0.16. We calculated the loss due to selection, and we said that there was no loss in the homozygous dominants or the heterozygous because there was no because they have a fitness of 1, and we said that the loss due to in the homozygous recessive genotype was 0.04, bringing us a new frequency of 0.12 for the homozygous recessives. We note that 0.36, 0.48, and 0.12 do not add up to 100. It adds up to 0.96. So we need to create corrected frequencies by dividing each genotype frequency by 0.96. So the homozygous dominant frequency was originally 0.36. We divided by 0.96. We note the frequency of the homozygous dominants in this population is now 0.375. The frequency of the homozygote, 0.48, divided by 0.96, is now 0.50. And the frequency of the homozygous recessive, 0.12 divided by 0.16 equals 0.125. These new corrected frequencies add up to 1. So the new allele frequencies for after one generation of selection, the frequency of the dominant allele, 0.375 plus 1/2 of 0.5 equals 0.675, and the frequency of the recessive allele is 0.125 plus 1/2 of 0.5 equals 0.375. These values right here of the 0.375, this is the frequency of the homozygous dominant. This 0.125 is the frequency of the homozygous recessive. This 0.5 in both situations is the frequency of the heterozygote. So what we're seeing is that whereas before the frequency of the dominant alleles was 0.5 and the frequency of the recessive allele was 0.5, after one generation of selection, A has increased and dominant A has increased, recessive A has decreased. If this continues over multiple generations, dominant A is going to increase towards 1, recessive A will decrease towards 0. Okay, let's look at a case of selection against recessive homozygotes. An example in humans of a case of selection against recessive homozygotes is a medical condition called Tay-Sachs disease. Tay-Sachs is caused by a metabolic disorder that results in blindness, mental retardation, destruction of the central nervous system. Children with Tay-Sachs generally die within the first years of life. They rarely live past 5. The disease is caused by a recessive allele and occurs in individuals who are homozygous. So generally you have normal parents but they happen to be carriers, and they have a child who is now homozygous. The heterozygotes, of course, carry the allele, but they are not affected. When deleterious alleles are recessive, such as with Tay-Sachs, the frequency of the recessive allele is generally not zero, because heterozygotes continue to pass the allele on from generation to generation with no impact on phenotype. Nonetheless, the frequency of the recessive allele is still going to be very low. This low frequency is maintained by mutation, but is kept from increasing by natural selection. So, selection against dominant alleles, let's consider this example. As an example, considering the starting point, as in the previous example, we have 50 individuals that are homozygous dominant, we have 100 individuals that are heterozygous, and we have 50 individuals that are homozygous recessive. The allele frequencies we're starting with are the dominant A is 0.5, recessive A is also 0.5. Let's complete, we have complete, in this example, we'll have complete selection against the dominant allele. And what this means is that any individual with a dominant allele is going to die. Okay, it's not going to survive and reproduce. So the fitness will be 0 for homozygous dominant genotypes and heterozygous genotypes. The fitness will be 100% for individuals that are homozygous recessive. So in this example with the P's, these 3 individuals would have a fitness of 0. This individual with the heterozygous genotype is going to have a fitness of 1. Okay. So let's look at selection against the dominant allele. We have our 3 genotypes: homozygous dominant, heterozygote, homozygous recessive. The homozygous dominant has a fitness of 0, heterozygote fitness of 0, because we're looking at selection against a dominant allele, and they both have dominant alleles. And then we have the homozygous recessive, has a fitness of 1 because it has no dominant alleles. There are 50, 150 individuals, 200 individuals total. We calculate the allele frequencies, the frequency of the dominant allele. We have 100 dominant alleles from homozygous dominant individuals. We have 100 dominant alleles from the heterozygote individuals and 100 from the recessive. And then we have from the homozygous recessive individuals, we have no dominant alleles, and we have 100 recessive alleles. There are 200 total A alleles, 200 total recessive alleles. 200 divided by 400 gives you P equals 0.5. 200 divided by 400 equals Q equals 0.5. Okay, so the next thing we need to do is we need to calculate the loss in genotype and allele frequencies due to selection. To calculate the loss due to selection, the first thing we do is we multiply the selection coefficient by genotype frequencies. Okay, so these are our genotypes: homozygous dominant, heterozygotes, homozygous recessive. We know the selection coefficient for the homozygous dominant must be 1 because fitness equals 0. We know the selection coefficient for the heterozygote has to be 1 because fitness is 0. We know the selection coefficient for the homozygous recessive has to be 0 because fitness is 1. These are our genotype frequencies that we've calculated from our P's and Q's. And now we multiply our selection coefficient by our genotype frequencies. 1 times 0.36 equals 0.36. 1 times 0.48 equals 0.48. 0 times 0.16 equals 0. So the loss of the homozygous dominant is going to be 0.36. The loss of the heterozygote is going to be 0.48. The loss of the homozygous recessive is going to be 0. We now have to create corrected frequencies. We now have 0.36 minus 0.36 is 0. 0.48 minus 0.48 is 0. So these are the frequencies of these genotypes after 1 generation of selection. And the homozygous recessive, 0.16 minus 0 is 0.16. They all add 0 plus 0 plus 0.16 equals 0.16. It doesn't add up to 1. We divide these all by 0.16. 0 divided by 0.16 is 0. 0 divided by 0.16 is 0. 0.16 divided by 0.16 is 1. So the frequency of the homozygous recessives is now 1. So we now calculate the new allele frequencies in the population after one generation of selection against dominant alleles. A equals 0, because this is the frequency of the dominant allele of the homozygous dominance, plus 0 divided by 2, because the 0 is the frequency of the heterozygote. Your frequency of the dominant allele is 0. The frequency of the recessive allele is 1, because it's the frequency of the homozygous recessive genotype, plus 0, because this is the frequency of the heterozygous genotype. So what we see is A has decreased, and dominant A has decreased, recessive A has increased. Actually, you wouldn't even just say dominant A has decreased, we'd say dominant A has been eliminated. The dominant allele has been eliminated after one generation because the selection coefficient was 1. No further change will occur in this population unless the allele, the dominant allele, is reintroduced into the population through mutation or migration. Let's look at another less severe example of selection against dominant alleles. In this example, we see that we have 3 genotypes once again: homozygous dominant, heterozygote, and homozygous recessive. The fitness of the homozygous recessive is still 1, but the fitness of the homozygous dominant and the heterozygotes is less than 1. It's not 0, it's 0.75. The selection coefficients, therefore, are 0.25, 0.25, and 0. These are our genotype frequencies. Okay, we calculate our dominant allele frequency, 0.36 plus 1/2 of 0.48 is 0.6. The recessive allele, 0.16 plus 0.48 divided by 2 equals 0.4. So the P equals 0.6, Q equals 0.4. Okay. We now want to calculate the loss due to selection. We're going to multiply once again the selection coefficients by the genotype frequencies. These are our selection coefficients, and once again they are the opposite of the fitness, the inverse of the fitness. We know that the fitness of the homozygous recessive is 1, so it's 0. The fitness of the heterozygote was 0.75, selection coefficients 0.25. The selection coefficient of the homozygous dominant is 0.25 because fitness was 0.75. We now calculate the loss due to one generation of selection. We multiply the genotype frequency by the selection coefficient. 0.25 times 0.36 equals 0.09. The loss for the heterozygote is 0.25 times 0.48 equals 0.12, and the loss of the homozygous recessive is 0 times 0.16, which equals 0. So let's calculate this out. We have we have our 3 genotypes. We have our original genotype frequencies. We have the loss due to selection right here. So 0.36 minus 0.09, our new frequency of the homozygous dominant genotype is 0.27. I'm sorry, I didn't mean to go forward. The new genotype, the heterozygous genotype, the original frequency was 0.48. The loss due to selection is 0.12, so the new frequency is 0.36. For the homozygous recessive, it was originally 0.16. We had 0 due to loss due to selection, so the new frequency is 0.16. But we add up 0.27, 0.36, and 0.16, and it only adds up to 0.79. So we need to correct to 100. So 0.27 divided by 0.79 equals 0.34. So the new frequency of the homozygous dominant is 0.34. The new frequency of the heterozygote, 0.36 divided by 0.79 equals 0.46. And the new frequency of the homozygous recessive equals 0.20. Okay. So the new allele frequencies based on, let me go back to the previous slide, based on these new corrected frequencies, okay, 0.34, 0.46, and 0.20 are now calculated. The dominant allele is homozygous dominant allele frequency is 0.34. The heterozygote is 0.46. So the frequency of the dominant allele is 0.34 plus 1/2 of 0.46 equals 0.57. The frequency of the recessive allele is 0.20 plus 1/2 of 0.43 equals 0.43. So what we're seeing is the dominant allele has decreased slightly from 0.6 and the recessive allele has increased slightly from 0.4 due to minor differences in the fitness between dominants and recessives. This is due to some selection against the dominant allele. There are examples of these types of dominant alleles in humans where we do see selection against dominant alleles. An example of a dominant allele in human populations is a chondroplastic dwarfism. This type of dwarfism results in small size and abnormal body proportions. It's caused by a dominant allele, a rare dominant allele, that's found in very low frequencies in human populations, 0.00005, very, very low. Because the allele is dominant, individuals with one or two of the alleles show the condition. So that means that virtually all dwarfs are heterozygous. This condition is usually caused by a mutation that occurs in the sex cells of the parents, and we know this because 80% of the dwarfs are born to heterozygous normal parents. Okay, so it usually occurs in a mutation, and it usually found 80% of dwarfs have two normal parents, because if the parents were heterozygous, they would have one of those dominant alleles and they would exhibit the condition. So we know that 80% of dwarfs have two normal parents. In cases where dwarfs mate, the offspring can be either homozygous for the disease, and they generally die before or shortly after birth. The low frequency of this condition is the result of natural selection acting to remove the harmful allele from the population. Although there is no major risk or mortality for a heterozygous condition, selection acts on differential reproduction. The third type of natural selection, that way that natural selection operates on allele frequencies, is selection for the heterozygote. Selection for the heterozygote is often referred to as selection against the homozygote. Selection against the homozygotes increases the frequency of one allele and decreases the frequency of other alleles. And over time, allele frequencies are expected to approach 0 or 1, depending on which allele is selected for and which allele is selected against. So we might expect that most populations have allele frequencies either close to 0 or 1, and few populations with intermediate values. But what we see is that studies of human genetic variation demonstrate that most traits, allele frequencies, have intermediate values, values such as 0.3, 0.5, 0.8. Why is this the case? Some people have suggested that the reason we see so many intermediary values of these allele frequencies is that natural selection is not yet complete, and over longer periods of time, the allele frequencies would move closer to 0 and 1. Populations at 0.3 would move towards 1, populations at 0.8 would move towards 1. The majority of information regarding allele frequencies in human groups makes this very unlikely. So the question is, why do so many intermediary alleles show intermediary allele frequencies? is there a way that natural selection can produce intermediary allele frequencies? A classic example of intermediate allele frequencies in humans' populations can be seen with a disease called sickle cell anemia. Sickle cell anemia is a disease that results from a single amino acid substitution, a mutation. It's called a point mutation. It's at position 6 within the hemoglobin beta chain. And this hemoglobin beta chain has 146 amino acids. And it is only at position 6 that there's a single change in the amino acids. For example, look at the following graph, at the following figure. This is normal. This is the first 1, 2, 3, 4, 5, 6, the first 7 amino acids in the hemoglobin chain, okay? And this is what this is the 3 bases. We see CAC, GTG, GAC, TGA, GGA, CTC, and CTC. And they code for these specific amino acids, okay? When an individual has sickle cell anemia, they have the first 5 amino acids are exactly the same as what we see in people with normal hemoglobin. However, position number 6 has a single change. Instead of CTC, it has CAC. So instead of this amino acid, it produces this amino acid. Okay. Now, why is this relevant at all? Well, hemoglobin is a protein found in our red blood cells, and it transports oxygen from our lungs to the various tissues in the body. And every red blood cell contains millions of hemoglobin molecules. In sickle cell anemia, what happens is abnormal hemoglobin molecules pack together to form rods. This causes the red blood cells to become crescent or sickle shaped. So you can see that a normal red blood cell looks like this. A sickle red blood cell looks like this. These deformed blood cells, they break very easily. They're often removed from circulation, which produces anemia because you no longer have enough protein in your blood. And these sickled cells also result in clogged blood vessels. Individuals that are homozygous for this mutated allele, and we say they have HBS, are likely to die very, very early in life. Even in the United States, with aggressive medical intervention, life expectancy for individuals with sickle cell anemia is often less than 30 years. So on the surface, this appears to be a classic example of selection against the homozygote. You don't want individuals to have both alleles for sickle cell anemia because then you'll exhibit it. So the expectation would be that sickle cell allele frequencies should be close to 0. However, we find that many populations do have sickle cell frequencies near or close to 0. However, there are a number of populations in Africa, in India, and in the Mediterranean that have much higher frequencies. Some West African groups, the frequency of sickle cell alleles is greater than 20%. In Gambia, in West Africa, 40% of the population is heterozygous. That means 40% of the population has at least one sickle cell allele. So the question is, how can these harmful allele frequencies exist at such high frequencies? The answer to that question is the form of a selection known as selection for the heterozygote. The way that selection maintains very harmful alleles at high frequency is selection for the heterozygote. And when you have selection for the heterozygote, you're selecting against the homozygote. Let's consider the following example. We have the following 3 fitness values. We have 3 genotypes: homozygous dominant, heterozygote, homozygous recessive. 70% of the population has homozygous dominant. 70% The fitness for the homozygous dominant is 70%, the fitness for the heterozygote is 100%, and the fitness for the homozygous recessive is 20%. What this means is that 70% of the people that have homozygous dominant alleles survive, and 20% of the homozygous recessive alleles survive for every 100 people with the heterozygous genotype. This is selection for the heterozygote and selection against the homozygote. So let's look at it in this manner. We have our three genotypes: homozygous dominant, heterozygote, homozygous recessive. We have our fitness values already, 0.7, 1, and 0.2 for the three genotypes respectively, from which we can calculate our selection coefficient. When the fitness is 0.7, selection is 0.3, fitness is 1, selection is 0, fitness is 2.0, selection is 0.8. We know our genotype frequencies. The frequency of the homozygous dominant is 0.25, the frequency of the heterozygote is 0.5, the frequency of the homozygous recessive is 0.25, from which we can calculate our allele frequencies. And we note that the allele frequency for the dominant allele A is 0.5, so P equals 0.5. Q, the frequency of the recessive allele, also equals 0.5. Okay, so what happens before selection? In the first population, we have 200 individuals, and before selection, we have 50 individuals that are homozygous dominant, we have 100 individuals that are heterozygous, and we have 50 individuals that are homozygous recessive. After selection, the allele frequencies are going to be the dominant allele will have 170 divided by 290 equals 0.586. The recessive allele will have 120 divided by 290 equals 0.414. So how did we calculate that? To calculate the loss due to selection after one generation, we have our selection coefficients from the previous slide, and we have our genotype frequencies. We now we multiply the selection coefficient by the genotype. 0.3 times 0.25 equals loss of 0.08. For the heterozygote, 0 times 0.50, we have a loss of 0. For the homozygous recessive, 0.8 times 0.25 equals 0.20 is our loss. So those are our losses. We now calculate subtract the original frequency, the loss from the original frequency. The original frequency of the homozygous dominant was 0.25 minus the loss due to one generation of selection is 0.08 equals 0.17 is the new frequency. 0.150 minus 0 is 0.50. And for the homozygous recessive, we started with 0.25, we lost 0.20, so our new frequency is 0.05. 0.17, 0.50, and 0.05 equal 0.72. So we need to create corrected frequencies. 0.17 divided by 0.72 is 0.24. 0.50 divided by 0.72 is 0.69. 0.05 divided by 0.072 is 0.07. So our new frequencies are 0.24, 0.69, 0.07, and those are the new frequencies. So we originally had 0.25 for homozygous dominant, we now have 0.24. We originally had 0.50 for the heterozygote, we now have 0.69. We originally had 0.25 for the homozygous recessive, we now have 0.07. We then take these values, let me go back, the 0.25, 0.69, and 0.07, and we calculate our new allele frequencies, the ones I showed you before. We have 0.24 plus 1/2 of 0.69 divided by 2 equals 0.585. The frequency of the recessive allele is 0.07 plus 0.069 divided by 2 equals 0.415. So what we're seeing is the frequency of the dominant allele has increased and the recessive allele has decreased. The question is, why has that occurred? Selection for the heterozygote, both alleles are being selected for, both the dominant allele and the recessive allele. But what we're seeing is that every heterozygous individual is still contributing alleles to the next generation. Also, both alleles are being selected against. The heterozygotes are being selected against, and homozygous recessive people die and fail to reproduce. So we're seeing selection against the homozygotes involves selection for and against both alleles. Because the fitness of homozygous recessive is greater in this example than the fitness of the heterozygote, proportionally more individuals with homozygous dominant will survive than homozygous recessive individuals. So there'll be more dominant A alleles in the next generation. Eventually, a balance will be reached between selection for and against the two alleles. The exact value of this balancing point will depend on the fitness of the homozygous genotypes. Selection for heterozygotes is often referred to, selection for the heterozygotes is often referred to as balancing selection. Given this model, the distribution of sickle cell anemia frequencies makes sense. In all environments, there is selection against the sickle cell homozygote because people die before reproducing and it has a low frequency. However, when malaria is common, in environments where malaria is common, the heterozygotes have an advantage because they are less susceptible to malaria. Malaria is a disease caused by a protozoan parasite predominantly called Plasmodium falciparum, although there are other varieties of it. This disease is transmitted to humans by infected mosquitoes, and affected individuals suffer recurring episodes of illness throughout the life. They're also more likely to contract other diseases, often fatal diseases. So what we say is that malaria victims have a reduced fitness. There are more than 2 million people that die from malaria every year, and more than 3 million people are infected by malaria each year. Prior to ethics, there was a very interesting study that was conducted on malaria. It was this guy named Allison. And he conducted an experiment on 30 volunteers from a village called Luo in West Africa. He injected malaria into patients with the following genotypes: HBs, so they're homozygous recessive for the genotype, for the sickle cell anemia, HB, so they are heterozygote, and HB, so they're homozygous normal for not having malaria. And what he did was he injected malaria into patients that had those three genotypes, and the results were very interesting. None of the individuals that were homozygous for sickle cell anemia developed malaria. They all developed sickle cell anemia, but none of them had malaria. 1 in 10 individuals that was heterozygous with the HB genotype developed malaria, and all of the individuals with the non-sickle cell anemia hemoglobin genotype developed malaria. These results are very instructive. Basically, they suggest that mutated hemoglobin confers some protection against malaria. This is expected because once an individual is bitten by a Plasmodium parasite, the parasite sets up residence in the red blood cells and eventually goes through a developmental process that destroys the cell. Cells containing sickle cell hemoglobin are not conducive to the parasite's development, making heterozygotes less susceptible to malaria. Research demonstrates that parasite infects the red blood cells of heterozygotes, but when the sickle cell begins to sickle, it develops that sickle shape instead of that nice round plump shape, the potassium levels in that cell drop and the parasite dies. So basically what we see is that homozygotes for sickle cell anemia allele die. Homozygotes for normal hemoglobin are likely to get malaria and possibly die. And so what we're seeing is selection against both homozygotes. You don't want to be homozygote for sickle cell anemia, and you don't want to be homozygote and not have a sickle cell allele, because then you're going to get malaria. So we're seeing selection for the heterozygote. A balance of sickle and non-sickle cell allele frequencies has been found in many of these human populations. We can calculate equilibrium frequencies from the genotype fitness using the following formula. S1 p hat equals S3 q hat. So S1 is the selection coefficient in population 1. P hat is the frequency of the genotype frequency in the equilibrium population. S3 is the equilibrium frequency for the dominant allele. S3 is the selection coefficient for the third genotype, and Q hat equals the equilibrium frequency for the recessive allele. Now we really don't want this formula, we want this one where we're solving for P hat and Q hat. What is the frequency what is the equilibrium frequency for the dominant allele? What's the equilibrium frequency for the recessive allele? And we can calculate this. You don't need to know how we go from here to here, but we can calculate it from S3 divided by S1 plus S3. These are your selection coefficients, and the frequency for Q hat is S1 over S1 plus S3. And once again, so we can calculate equilibrium frequencies from selection coefficients. Equilibrium occurs when S1 P hat equals S3 Q hat. And this equilibrium represents a balance between selection against the two alleles. When you have a stable equilibrium, there's going to be no further changes in those gene frequencies or allele frequencies. So let's look at a specific example because it's easier to understand. We have our three genotypes: homozygous dominant, heterozygote, and homozygous recessive. We know what our fitness is. Okay, the fitness of the heterozygote is 1, the fitness of the homozygous dominant is 0.75, and the fitness of the homozygous recessive is 0. From that, we can calculate what our selection coefficients are. Fitness is 0.75, the selection coefficient is 0.25. Fitness is 1, selection is 0. Fitness is 0, selection is 1. We now calculate our value for what the equilibrium frequency for the dominant allele and the equilibrium frequency for the recessive allele. So S3 equals 1 divided by S1 plus S3, so that's 0.25 plus 1. 1 divided by 1.25 equals 0.8. So the equilibrium frequency of the dominant allele is going to be 0.8. The equilibrium frequency of the recessive allele in this example will be 0.2. What this means is at these equilibrium frequencies, the gene frequencies will remain the same generation after generation, regardless of the strength of natural selection. We're going to start at P equals 0.8 and Q equals 0.2. And let's test this out. We have our original genotype frequencies, okay, and we see loss due to selection, okay. We calculate 0.25 times 0.64. Our new frequency equals 0.16. We correct it to 0.48. We subtract it and we get 0.48. And then we have 0.48 divided by 8 equals 0.6. And then this is 0.4. And then the frequency of the homozygous recessive is 0. We now calculate our new allele frequencies, even though we've had another generation of selection. A equals 0.60. Okay, because that's the frequency of the dominant alleles, plus 1/2 of the heterozygote. 0.4 divided by 2 is still 0.8. Frequency of the recessive allele, 0 plus 1/2 of 0.4 is 0.2. So even though we've had some really strong selection, especially against these homozygous recessives and the homozygous dominants, the allele frequencies are not going to change because it is now at an equilibrium where the selection is operating on both of them and there's selection for the heterozygote. That's pretty cool, huh? This represents what we call a balanced polymorphism, selection for the homozygote. When you have selection for the heterozygote, you can use the formula S1 p hat equals S3 q hat to determine the selection coefficient of one homozygote when you know the equilibrium gene frequencies and selection coefficient of the other homozygote. So it's very easy to calculate out. Okay.